today.
Sure.
Okay, I got it. So first of all, I'm very happy to introduce our next speakers, María Harkiolaki. I don't know if I'm pronounced well that.
Very well indeed. I'm very pleased. Well done.
She is an associate professor of the Correlative Cryo-Imaging Beamline at the UK synchrotron diamond light source, which is the reference synchrotron in UK. Maria also was a principal investigator at the structural biology division of the Nuffield Department of Medicine at the University of Oxford, where her research focused on understanding the structure of molecular interactions that define antigen recognition and autoimmunity. Now her research focused on cutting-edge microscopy technology development, structural cell biology and morphogenesis pathways, and understanding host pathogen interaction at the cellular and molecular level, with emphasis in viral escape and clearance pathways. I have to say also that I'm sort of happy because I was contacted by someone that worked with Maria Chidina. He's also a research from the synchrotron diamond light source, and so we are trying to initiate a collaboration. And unfortunately, came COVID, so I have some thing to want to really pursue with this collaboration. Study with the changes produced by expression of connexin in many cells. And so, okay, Maria, I'm happy you can give us a talk about your research facility, but also about your own research. And so okay, I give your.
Thank you very much, Agustin. It's a real pleasure. I'm going to share my screen. So let's start. And there we go. All right, so I'm going to talk to you. This is a fairly long talk, but I'm hoping that by the end of it, you know, we're going to take some breaks and hoping that you'll appreciate what we do. So I'm going to talk about investigating the natural ultrastructure of cells using soft X-rays and laser light. And I am, as Agustin said, María Harkiolaki. I'm the principal Beamline Scientist of the Correlative Cryo-Imaging Beamline at Diamond B24. And we'll start by an introduction to diamond light source and the Beamline. So I'm never quite sure when I talk to new people if they're familiar with synchrotrons. So this is the titular sort of slide on synchrotrons. We're up here, very, very far away from you. And I wanted to make this point because although we're that far away, in fact, we have users and collaborators from across the world. If anything, the lockdown and COVID brought about a new age of remote access. And it's, in fact, the synchrotron in the UK is supremely accessible to anybody, especially academics from around the world. As I said, the synchrotron, the only thing that it is a particle accelerator. You have a linear accelerator. I'm showing the structure. This is diamond itself. In fact, the beamline is somewhere in here. So you normally have a linear accelerator producing all the electrons, which are fed into a booster ring. They're accelerated near to the speed of light and then fed into a storage ring. Now, while the electrons turn around and around at the storage ring, every time there's a turn, they produce X-rays. What effectively we do is we take these X-rays and we treat them as light, and we direct them to different microscopes. Effectively, each of these hutches called beamlines is a different microscope. Maybe they have different intensity. They could have slightly different wavelength, but all that they are is machines that look into matter. So for that reason, hence B24. Now, B24, our beamline, is within the biological cryo-imaging group at Diamond, and we're part of a collective with the electron microscopy group, and we have all the information here on our website. I have put a little QR code if you're interested, and then the people that you normally see as us. So you have the staff. Elias has left us now. Chidima Archana has recently started. So and we have our students and PDRAs. So I'll start by saying again, as I said, the beamline is a machine that harvests light from a synchrotron bending light. We have a number of mirrors, and all this light is directed to the microscope. Now, all these optics here, we have a condenser objective detector. There are special optics for X-rays. The Beamline has a substantial amount of automation, as you can see here. But what I tried to make a point here, this is the work of an awful lot of people. It's not just one group, it's an awful lot of people and a lot of technique development. So I will start with the obvious. Why is there a requirement for high resolution 3D cryo-imaging? And I believe we're all very familiar with this, in that as you image across scales for the biological sciences, different projects, we have different emphases. Some people will be looking at organisms, tissue, all the way to the high resolution and a high detail information of atoms and proteins. And for all of these, there's specialized techniques that are made. So we have computerized tomography, CT for organisms and tissue, and then fluorescence microscopy, all the way down to X-ray crystallography with an extreme resolution, 0.1 nanometers. And that's great. But every time you gain something in resolution, you lose context. You cannot, to date, it's near impossible to localize a single atom, a single drug, for example, or molecule or a protein within a huge organism. So you take, you take some liberties. You use methods that maybe bridge 2 or 3 of these techniques. And that's what we've done at B24. We've bridged the gap between effectively visible light microscopy and electron tomography. And we provide 2 methods that are complementary. And I'll explain a little bit how this works. But they're both honed on giving you images on cells, organelles, or macromolecular complexes. So in a nutshell, we do correlative imaging, but as a Beamline, we provide X-ray invisible light microscopies, our remit basically to develop and provide this. We develop protocols, automation, and aspire to user-friendly access. Our involvement starts at the experimental design, protocols, expertise, data collection, imaging, and even data correlation. Now, I need to explain also that we work in a facility. It's our job to develop these methods and provide them to users, but we have our own research programs. We have our own collaborators. So we're there for the experienced users. We're also there to start people anew in this method. And it's kind of important. We'll come to this slide one more time. But how this microscopy platform can help your research starts with, it needs to be the right project. So we can deal with that studies of molecular ultrastructure, chemical localization, cellular mechanisms, effects of drugs on cellular structure, steady state or induced organelle substructure, infection mechanisms at the cellular level, pathogen defense, development, you name it. It has to be, though, focused on the cell level. And then it needs to be the right method for the right project. What we do is super resolution microscopy, which means that we can't give you real life, real time imaging, because you would sacrifice the super resolution element of it. So it's a snapshot imaging. We plunge freeze and arrest cells through vitrification, and that's how we interrogate, we image them. You need 3D data. So, provided you need image or volume of images, that's excellent. Single cell populations, as I said, and you need to have an understanding that cells have personalities. In fact, when people start looking into cells, it is such a multi-parameter universe that within a cell population that starts from a single progenitor, you can have so much variation to the reactions to drugs, for example, or pathogens. So this needs to be an understanding that it's a game of numbers. You need to do statistics. And then it has to be the right time in the project. We offer access for initial stages when we're exploring something, but really this is perfect for a mature project. Somebody who knows what they want, and ideally associated reagents. So if there's already fluorescently labeled cells, for example, that's perfect. All right, so with that in mind, I'll say that we have 2 major high-end microscopes. We have X-ray microscopy. You can see the microscope here with Nina and Adam, 2 members of our team. And you can see the fluorescent microscope, which is a structural illumination microscope. Both of these give you information, chemical localization of molecules or ultrastructure of the cell parameters. Now, these 2 images, these 2 microscopes are embedded within a platform at B24. And that means that we've taken the process from the very beginning, from cell culture, live cell imaging, all the way to light microscopy, cryosim, structural illumination. SXT is the X-ray microscopy that we do, all the way into processing of the data and understanding the results. And we've worked on all of these, and we've produced protocols, and we have expertise in all of these steps. But I'll take a step back, and I said already we're looking at cells, but what is a sample? When I say a sample for B24, Our sample carriers are TM grids. So these are gold wafers, like the circular wafers of about 3 millimeters, and you can see one against a 5p coin here. And with the universal scale of a glass cover slip, you can see how big they are. And these normally have some assignment of area, so they're finder grids. And they normally have a thin film of carbon, which is used as a support for the cells. Quite often in the images that I'm going to show you, you'll see a surface full of very perfect holes. This is because this film is perforated and allows us to draw liquid through it. So you can see a schematic here of cells being deposited on a carbon foil, as I said, and then the grid. And of course, the cells in bright field, as we'll see in a little bit, don't look very amazing. They're like little jellies, but fluorescence gives us more information. And there are ways to get good samples and bad samples. This here, for example, a single cell on a grid is probably a most unhappy sample because cells like to be in clusters. But then if you overdo it, the imaging is going to suffer. All right, so with that in mind, we'll go to the second one. We'll start with the actual imaging. So we'll start with water window soft X-ray imaging. And I'll point out that I've actually skipped the Cryo here just because historically the beamline was built as a soft X-ray tomography beamline, just this method. And as I said, this is how it looks in a schematic of our optics. But if you were to visit us, it looks a little bit like that. I have to admit, looking down at the microscope, this is the microscope back here. Looking the other way has changed completely. It's a proper wet lab now. It definitely is not as tidy as that. But this is the place where we work, and you can see us basically pretending to be super busy while somebody was taking a photo. All right, so I'll start with the X-ray tomography. It's a 3D imaging technique, as I said, and it gives you the ultrastructure of vitrified whole cells at near states. This is important. So vitrified, first of all, because we plunge freeze them. No ice is formed. We arrest them. We snap freeze them. So we capture the true landscape of the cell when at the moment that we want. And it's at near native because we don't need to fix, we don't need to stain, we don't need to treat it in any other way, and we keep them always hydrated. And what you can see here is actually our field of view, what we see, how much we can see. There are two cells here. There's a nucleus top left, and there's a nucleus right at the bottom. And these two cells are in contact here on the side. And each nucleus that you see here has lots of dark spots. These are actually herpes virus capsids. It's one of my students' project. And you can give you an idea of the resolution you can expect. And of course, this is imaging through the volume of a fully vitrified and fully infected, in this case, cell. We achieve this contrast by using a special kind of wavelength. So the X-rays that we use are called soft X-rays. They don't travel much in S. All of our equipment are in vacuum. But what they do is they get absorbed preferentially by carbon-rich biological matter, especially when it's structured like vesicles or membranes, and it doesn't get absorbed as much but by the oxygen-rich media that's around it. So we get natural contrast. It's limited angle tomography.
I'm sorry,
Yeah.
Maria, can I have a question?
Yeah.
I'm sorry to interrupt you. Is this resolution is for the axial resolution, or is the lateral resolution? Or...
We do have, yeah, the lateral is always diminished because you lose a little bit, but in theory, you should be able to achieve 25, theoretically 25 nanometers. We have measured up to 30 nanometers, but there is always an elongation factor along the direction of the beam, and that's because of the missing wedge. But the tomography itself, the same as the CT tomography you get in a clinical setting, has exactly, cubic voxels. So it's 25 by 25 by 25, yeah?
And how thick can be the sample?
Yeah, so they can be up to 12 microns thick. Most cells, when they're grown on a flat surface, will be, maybe 5, 6, 7 microns thick. So we can easily see 1 or 2 layers of cells. So the tomography, because we mentioned it, it's limited angle, which means we always have a missing wedge. It's the same as the electron tomography method that is being used. But the resolution, as I said, we have measured it. So this is the method. It is complementary to other imaging data, which means that the sample could have been imaged fluorescently and then given to us, or after us, there's other methods, and I'll get to that. And we have some scheduled upgrades. So I will move to the next step and say, when we start taking data, we start with projection X-ray data. I'll point out again that what you're seeing here is a snapshot that shows several cells. We're looking at several microns across. And of course, the snapshot is just a projection, so we don't get the full resolution here, but this was a pretty good image, so I thought I'd share it with you. These are A549 lung epithelial cells. And remember, no staining, just soft X-rays going through a fully hydrated sample. So you get an idea of the features that you can see without any enhancement in resolution. So when we collect data, we start with an image like that, and I've used here another mosaic. So if you see, these are repeat of squares, and this is our field of view. This is how much we can see at any single time. So we raster it, so we move the sample across and we get an overview. Then we focus on an area of interest. Here, in this case, it's here. These are U2S cells, by the way. These are, again, human cells. And the kind of data we collect looks like this. So this is our raw data. This is snapshots of the sample moving around a rotation axis at different angles. So we're imaging very much like you would do, like in the movie, the Matrix, basically. You image at different angles, put it together. And the effect that has is when you do put it all together and you reconstruct it, that data is now vanished. That data now looks like this. So we now have reconstructed it from a series of projections into a full volume. So if you see this volume now, you can see the nucleus on this side. We're seeing these holes are the perforated film that's holding the cells, and we're going back inside the cell. You can see all the mitochondria, the cristae, the ER, and all the membranes that surround them. These are all endosomes and multivesicular bodies, the digestive system of the cell effectively. And at the very top, these are gold fiducials that we use for alignment. So this is the data that we get. And depending on the optics we use, we have two zone plates of 40 nanometer objectives, effectively 40 nanometer and 25 nanometer. And the 40 nanometer gives us a good almost 20 microns area to look at, so much more. But then the 25 nanometer gives us a lot more detail. So if you see here, these are two cells infected with reovirus. In this image at 25 nanometers, you can actually see reoviruses in the cytoplasm of a cell, and I will stop it when we get there. The nucleus is here, and these are the viral factories. Now, reovirus is in the region of 80 to 90 nanometers, so it gives you a good idea of the resolution, also the real resolution in the cell. I like to show this because we collected this within one afternoon, and I need to note that data collection for us, this took 5 minutes to collect, and then auto-process, so we didn't have to do much. But then the group that did that as a test had already done some scanning electron microscopy on it, and it took them 2 years to get to the same thing. So the postdoc was not very happy at the time, but that proves the usefulness of this method. Okay. Also, if 10 or 16 micrometers are not enough, you can collect adjacent boxes and put them all together in silico at a later stage, which is what we've done here. You can see some human fibroblasts effectively. So the volumes that you accumulate can be stitched together. All right, so just to show you some of the signs that we've had using this method, and these are mostly published, so I won't stay on it, but collection of red blood cells filled with parasites, with Plasmodium parasites. And this is the raw data, and the process data looks a little bit like this. You can see it in obvious resolution gain, but also this group was very keen on figuring out how these parasites start rupturing membranes at the later stages of infection. So, and then the same group actually generated data on toxoplasma. And again, what you're seeing here is the process data, and it shows a parasitophorus vacuole with 4 parasites inside. And this is just a tiny part of a massive fibroblast that was holding this. And you can see the segmentation as it comes out. So this is admittedly larger structures. We recently had a publication showing a new cytotoxic organelle. So we were able to show that T cells produce SMAP, which are, these are particles that contain cytotoxic proteins. They deposit them on the surface of target cells and affect killing. So that was pretty nice to show. And I chose one of our projects at the Beamline because one of the major directions of my research is herpes viruses. So this is imaging in animal cells. We were looking at chicken cells with an animal institute here, and we were tracking the progress of Marek's disease virus inside the nucleus as well as outside in cytoplasm and the tunneling it produces. We were able to collect lots of wonderful data on different variants. Most of these, the strains that we look at in this case, were vaccine strains, so they're very important commercially. Okay, so soft X-ray tomography does not necessarily live in itself. And the problem with imaging is that what we capture depends on scale. This slide will mean absolutely probably nothing to most of you because it's based on a nursery rhyme that we have here about an old lady who swallowed a fly and then sent a spider to catch the fly, and etc., etc. So I'm just going to skip it very quickly. All I was trying to make a point is that if you can see image something at different layers, you get more information, okay? And I put together this slide just for you about 2 minutes before the presentation. All right, so what are the partners we have? I'll start with the most recent. The method plays very well with cryo-FibSem, so electron microscopy, or electron tomography. And we had a recent publication on SARS-CoV-2, well, recent, now 2 years, but anyway. And in which case, although they had very high resolution data, this was acquired laboriously and over many months, and it was few instances, whereas with our data, we were able to document a whole population of cells, give more of a statistical backing to the nuclear fragmentation that observed once. Then we were able to give statistics on alter organelles, what happens after infection is established, document cell death progression, and also document membrane surfing, which is has been seen with these viruses, where we could see the viruses sort of jumping through and surfing the membrane here. So all in all, our method, it's very quick, gives a lot of overview for any system. We've also partnered with X-ray fluorescence very successfully, and that's another project we had with looking into anti-cancer compounds and specific photoactivatable ones. So once data was collected showing the ultrastructural changes that these drugs cause, especially when they're photoactivated, but also in the resident state, which I'm not showing here exactly, but we could track the distribution of elements through X-ray fluorescence after that. So that worked really well also, another publication there. And of course, for us, the B24 correlative platform that we have on site at the Beamline uses cryostructural illumination microscopy as a perfect partner. And I'm bringing you back to this, so clearly I won't go through it again, but this is the fluorescence microscopy that I'm mentioning. In our case, it was very important that we could add chemical localization to be able to find the proteins that we're interested, or chemicals that we're interested in, this super detailed three-dimensional ultrastructure that we get from X-rays. So we worked to bring an imaging technique that could use the same sample. So the moment the sample, the cells are grown on a grid and they're plunge frozen, at that moment, we needed to make sure that they can go to each and every microscope so that we know that we're looking at the same area all the time. And the structural illumination microscope was done as a collaboration with Micron, that's the microscopy facility at Biochemistry and the University of Oxford. And Mick and Ian are the two lovely people that helped us. I tell a lie, Mick pretty much designed the whole thing, and Ian make sure that it worked. Ian is now at the Johns Hopkins, but he's still involved with the development. Now, structural illumination is an old technique, is nothing new. The idea is that you throw patterned light. So if you have a light, we use stripes, for example. If you use a light of a known pattern onto an unknown, basically your cells, for which you don't have information beyond the diffraction limit of the microscope, and then you study the interference patterns in Fourier space, you could extract these out more information than you could otherwise. So what we do is basically take, and this is conventional cryo-wide field, how it would look without any cryosim. Again, this is from my student project with HSV, so that's the human and envelope and mitochondria. The resolution, of course, I'm only showing 3D. Cryosim is super, breaks all the time, but that's our and then it can go through very thick samples, so it matches perfectly because it's vitrified, they can be thick, and once we finish, it actually is not damaging through a high fluorophores to blame on the information. So the dosage is, so the sample arrives at the microscope relatively. So and this will I'm a student, to prove the point, how an experiment runs at the Cryo-Sim. As I said, bright field, several cells here. We're looking into mouse cells, macrophages. Again, this is an immunity-related project of ours. Not very impressive. They look like little jellies, but the same microscope can give you a substantial information. And we label the mitochondria here. As I said, it was a training exercise, and you can see the difference from the diffraction-limited imaging here. So what the microscope can give you without any super resolution processing, and the reconstructed structural elimination data. So we're getting 2.5 times the resolution limit that we had before, plus we're getting confocal quality of imaging. So the same grid we put then in the X-ray microscope, and you can see here data acquisition. This is the raw data, one nucleus, another nucleus, lots and lots of organelles. These cells are really, really densely populated with organelles. And the little black spots, these are gold nanoparticles. We just use those for alignment. They have no biological relevance. Okay, when this is processed, the level of information you can get out of something like this is, to us at least, tremendous. You can see all the defined structures, the double membrane, you can see heterochromatin, nucleosomes, you name it. As I said, these cells are really, really pretty dense. And then you can bring the two together. And when you do that, and as I said, this was just an exercise for a student, but I'm just showing you what happens. If we couldn't identify the mitochondria, it would immediately tell us where they are effectively. And we've used that in many occasions to find the proper vesicles or the right structures that contain something we were tracking. Okay. So effectively, bring two things together, make the output so much more powerful than anything else. So the workflow, you can see it as a strip here. It's effectively the same workflow I showed you before. It involves some sample assessment and mapping. The Cryo-Sim comes next because X-rays are quite damaging to fluorophores. They do break atomic bonds, and then X-ray tomography follows. I need to point out also that after that, there's a substantial in silico path that we follow. We've managed to automate data processing, so reconstruction happens automatically. In most cases, if the fiducialization is poor, then you still need to do it manually. And we also have 3D correlation scripts. So I believe to the rest of the 3D volume imaging community, we're actually ever so slightly ahead in being very kind and very helpful to us and our users. The B24 correlative imaging platform, I wanted to share the first application we had. That's now a little bit old, it's 2 years old, but it was the first biomedical project we showcased and told the world about our method. And we were looking into reovirus. I showed you a little bit of the data before. Reoviruses are not nasty pathogens. In fact, they are vaccine candidates. They are potential, they have medical significance in cancer, and there are several trials around the world right now with these. It is also interesting to know the basic biology. These viruses, they're not enveloped, but to get into the cell, they tend to come in through clathrin-dependent endocytosis, which means that they get themselves into the cell, fabulous, but then they're trapped within a vesicle. So at the time when we were doing this work, it was obvious that they find a way to inject their RNA into the cytoplasm. And the idea from in vitro studies was that they possibly form pores, but nobody knew exactly how it happened. If the vesicles that contained them collapsed, nobody knew exactly when and how and where it happened. So we partnered with a group in Oxford and another one in Heidelberg at the time. And they were EM people, so they prepared the grids themselves. They looked beautiful, and this is bright field, just white light. And they did use fluorescence. So they had, the real virus was green with GFP, and they were using a galectin as a signal for the integrity of the vesicle. So if a vesicle had a leaky membrane, that meant that galectin would accumulate inside and it would fluoresce. I'll point out also that these grids were fully populated with cells, but not all of them had the endogenously expressed M-cherry galectin. So that's why in this case, you can only see one, but in fact, there's other cells around it. What we did, as I pointed out at the beginning, this is not a real-time imaging method. So we capture instances. So we had control. We prepared samples 1 hour post-infection, 2 hours post-infection, 4 hours post-infection. And in that time, we were able to show that by 1 hour post-infection, most of these little endosomes at the time, early endosomes, had leaky membranes. And then the population enlarged. And you can see here in this cell, it looks orange because you have co-localization of the green virus with the galectin signal. And then at 4 hours, in fact, you're looking at the recycling pathway of the cell, where quite a lot of, you have massive vesicles containing a lot of, well, effectively junk. And as an example, I'm showing you one of the 4 hours post-infection when you have the X-ray data and you superimpose the fluorescence. There was a huge curve in this cell, so we're only looking at a thin area. The nucleus is on the top here, and then you're seeing lots of vesicles. If we focus on this pretty big, large vesicle, this is a late endosome, most likely recycling, but we were able to localize very cleanly, that to decipher very cleanly from the galectin signal that these vesicles, this vesicle was leaky. So somehow all this galectin was managed to be siphoned in, but also contained the debris, basically, the remnants of the original virus capsids. So effectively, we were able to show not only that we use this method to mine the massive amount of information you get in X-ray or tomography and localize our field of view and know where to look to find the virus, but at the same time, we're able to show that the virus comes out of these vesicles, injects this RNA very early on, within half an hour to an hour, but leaves the vesicles intact, which is a way of hiding also the fact that there's an infection going on. So architecturally, the cell has no way of knowing that something horrible is happening. Anyway, so that was our small contribution in that field. And of course, this is from the 2 hours post-infection, because we had lovely vesicles at the time, just to show you how much you augment the information content by having the fluorescence. So you don't have to look over and over all the vesicles, you know exactly where to look for your virus. All right. Of course, we have challenging subjects. For example, although we get really very good structural information on carbon-dense qualities, actin is actually, although structured, doesn't have a lot of carbon. They're fine filaments, so we use the fluorescence to sort of find vesicles that were covered with actin or networks of actin collecting different vesicles. So we used, it was a lovely complementation of the X-ray and the fluorescence. We have projects, for example, here I wanted to show you this data we collected on a primary control site. We knew there was a cilium there, but regardless of the high amount of information, and I've labeled the mitochondria here, it was difficult to find the actual structure of the cilium. However, let's stop this one. Once you had the fluorescence superimposed, and then we're coming through the same cell, it's very obvious where to look to decipher the information we wanted, effectively the cilium, as you see here. All right, so another one of our projects, it's very useful. The fluorescence in X-ray is fantastic for isolating and identifying protein islands in cells. So one of our projects is looking into influenza biology. So we were tracking the accumulation of viral proteins in distinct islands within the cytoplasm. These are membraneless, so very difficult to track. However, with a fluorescent signal, we could track them relatively easily. So we're hoping to be writing this up very soon. We've used the correlative method for industrial applications. So we partner with the industry. Industry does have to pay to access us, but in this case, we did a proof of principle. We were looking into active virusomes, effectively exosomes that are loaded with antigens and genetic material. In this case was Zika and chikungunya, and we were able to show not only the distribution of these vesicles and their properties, but also how well the cells, the cells in a cell culture fared upon exposure. Because after 2 hours, we knew that after a day, the cells really struggled to recover, but within 2 days, they had recovered. Cell cultures can be a very sensitive indicator if something is toxic or not. So that was very encouraging that they were able to recover. Not only that, but after a few days, we were able to show secondary expression and confirm what the company was proposing, that the cells were able to accumulate the DNA, the plasmids they were providing, and actually start producing their own antigens. So I'll reiterate now that you've seen it. the requirement for our methods, and I've shown you cells and organelles. And I will go back, which I hope now makes a little bit more sense, about the right focus of a project. So as you've seen now, I've shown you that anything that happens in a cell, if you're interested in a structural, architectural, or chemical side of it, then these are the methods ideal for that. It has to be the right method. I talked again and again about snapshot imaging, 3D data, and it has to be the right time. So as I said, we always make time for new and developing projects, but really this is perfect for a group that already has a good question and good reagents. And before I'm almost at the end now, but I wanted to make sure that from the whole workflow, and I'll say again, I make a huge disservice to the people that have put a lot of time to make the in silico processing, the later on after data collection, easy. And that was a massive amount of work. The most important place is sample preparation. We've written a Nature protocols on that. I could not possibly emphasize enough how important it is to prepare good samples for a project to succeed. So that's very, very important. And we're always there to help, we're there to collaborate, but we're also there to just give advice. All right, so transparency and accessibility. Our Beamline, who knows about it? So we have a playlist within the YouTube channel of Diamond, so you can find a lot of videos there that explain the technique, that explain what happens at the Beamline, that teach our users how to do things. And then we do have a Twitter profile, so I'm hoping by the end of this, all of you will be following us. It's B24 Light. And our website is, I mean, we spend a lot of time keeping it up to date and making sure that it has all the information that even a novice user will need. We publish as much as we can, and every publication we have is open access, and most of our code is on GitHub. We also do prepare conferences, take part in conferences. We run our own workshops, so we publicize them in the Diamond website and also in our Twitter site. And of course, we have our collaborations. So I'll be lying if I said we're the only Beamline doing it. Right now, you have several Beamlines that have some form, that offer this technique in some form or another. Some are in development mode, some have been there for a long time. The two that are very, very similar is the one in Spain and one in the UK. We offer the Cryo for now, at least, is just sourced at our side. Okay, I have to say that clearly I'm a little bit biased, but Diamond is B24 is the best. You don't tell that to anybody else. And who can access? We have users from around Europe and around the world. None from your side of the world, so we're hoping to remedy that. We give most of the user time, most of the X-ray time to our users, and this is our allocation. And this is a very typical shift allocation for a synchrotron. If you go to the website, you can find more about that, but effectively, all access is given via applications. People submit proposals, and you effectively get a grant. And that proposal awards shifts. These relate to a huge amount of money because to access the facility is quite expensive. This is given for free in the proposals that are awarded time. There's two ways to get time: through a standard proposal that runs twice a year, or rapid proposal, which come to me, but normally this is just for proof of principle, you know, beginning preliminary. And these are all peer reviewed. So once it gets peer reviewed, it is an award, very much a legitimate academic award. And both microscopes have their own. Yes.
Valeria Silva wants to make you a question.
Yes, please.
Valeria?
Oh, thank you. I was going to wait until then, but it's okay. Maria, thank you for the talk. It's pretty cool what you do. So I have a question because I can see that the most challenging phase is the sample. So do you think it's viable to do this in super tiny slice of brain eventually? Like, are you working in something like that?
So we work, in fact, we have done brain slices. It's very, very difficult. Do you know why? The problem is that you can't slice it thick enough. Okay, so right now to slice a cryopreserved, you know, frozen slice, you would never be able to do below 50 microns, I think. Even that is a bit of a push. To get 10 to 20 microns, which is the penetration depth of our beam, To my knowledge, it hasn't been done yet because it's shears, you know, it's very, very difficult. I believe people have done 0.7 microns or something, so you can do a very thin slice, you know, and you do need to sort of permeate the tissue. You can't, we've been trying, yeah. So in theory, if somebody could produce something that's 10 to 20 microns thick, we can do it. Okay, so we were looking at brain tissue, and on some areas, because the cutting wasn't straight, you know, we could get some information. But the problem with your tissue also, it's localization. You need to find some way of fluorescently tagging what you want to see, because a lot of the material is going to be, it's dense, yeah. What we have done trying to sort of overcome this, and we're running now pilots with the MRC Mouse Institute. We take tissue samples and we disperse the cells, allow them to settle, and then we sort of examine the populations. You know, it's a pseudo tissue, it's not 100%. At the same time, we're developing biomatrices, so they have the 3D option, but it's still work in progress. Is that okay?
Yeah, super cool. We're waiting for
I'm almost
that.
done anyway. Okay, so you caught me just before the acknowledgements, just to say that this is the work of a lot of people, and I mean a lot of people. It's not just the technical people, our collaborators, other synchrotrons around the world, microscopy experts. It's definitely our users, because I have to admit, to develop all this, we made our users work for us at some point. Okay, it's a bit shameless, but we had to do it. So yeah, that's it, I guess. Thank you for listening. Questions?
I'm sorry. Thank you, Maria. I'm really excited about your talk. I mean, it's I can see a lot of possibilities, and of course, I mean, I have sort of the same question as Valeria asked you before, and I can see that, well, it is more suitable for tissue culture. Like I don't know, if you want to study, I guess, dendritic spines or something like that in culture, that could be possible.
We have done a lot of work with a couple of groups on neurons. That we have one that was focusing on Alzheimer's. So I can tell you that we've recovered neurons, so we put them on feeder layers. It takes a long while, and most of the time doesn't work. They don't recover their physiology as you know, they don't look as happy as I would like them to, but we have worked with those, yes. Okay.
Oliver has a question.
Yeah, thank you for your talk, Maria. Very impressive. I'm a bit jealous, or very jealous, actually. So to get this right, this is unfixed, yes, hydrated, frozen tissue. And how exactly is it frozen?
Plunge freezing. So we can use either plunge freezing or high pressure freezing. It's very similar to the electron tomography work, if you have any institutes around. We do use slightly modified techniques, but effectively the cells are grown on these grids, and then we use a plunge freezer. So you blot very lightly. So you take just the outer layer of water. You don't want to, you know, dry them or anything, and they go very fast into liquid ethane. From then on, they remain in liquid nitrogen or cooled by liquid nitrogen temperatures.
Okay, so no cryoprotection necessary.
No, cells have a natural cryoprotection, it turns out. Coming from a crystallography field myself, it felt unusual, but yeah, no, absolutely. If you keep them at a reasonable density of above 60%, they experience very little disruption. And to be honest, from the electron tomography people that sent us very, very dry, very highly blotted grids, they're very resilient.
Mm-hmm.
Thank you.
So the challenge, of course, also is the contrast, no? How I guess people is trying to do something just to pre-stain the cells to increase the. We don't need to stain.
Well, X-ray tomography, you don't stain the cells. The X-rays are you get contrast because of the natural absorption of X-rays. It's the same as CT in a medical setting, you know, for the body. We do exactly the same, but we do it for cells.
Yeah, I understood that, but I thought that one of the, challenge of this methodology is to increase the contrast of the very beginning. So I know that you can have the contrast naturally, the natural properties of the samples. Yeah. But can you also please staining with something that, because I know that you have to combine fluorescent microscopy with this.
No, I see what you mean.
So is it possible at the very beginning to do something like, some protein or something?
To get the ultrastructure, we hands down do not need to do that. And most of the contrasting agents you would use at that stage would damage the physiological presentation of the cells. We know because we give them a lot of drugs, in the projects. But you don't need to enhance this. What you're seeing here as contrast is completely unenhanced. If you want the chemical localization, if you want to assign localization for your protein of interest, then you really need to think about how can you fluorescently label it. That's when the contrast comes into mind, but not for the method. This method is very good at giving us contrast by itself. So we don't touch the cells. We do use an awful lot of cell membrane permeable dyes if we want to label mitochondrial lysosomes and the type. And that is basically for localization and identification purposes, not contrasting.
Okay, I can see that. Valeria.
Yeah, I have another question, but it's super ignorant. So you show this so huge picture about the place, and also you show this map with collaboration. And my question is how it works. So people who are working in viruses, they do everything at their lab, and then they do the vitrification and they send to you, or you have many facilities all around your place?
No. So it depends on how our users, at what expertise they have. For example, the lab in Japan sent us one of their students. He prepared samples with us. We have all the wet lab facilities and all the cryopreservation and everything. And then once they done the first round, he went back. And because most universities and institutes, there will be somewhere a plunge freezer, it's so the sort of thing that most universities will have one or two. Cryoprotection and having cryopreserved samples is a major hassle for most labs, people don't just have liquid nitrogen lying about. So we either collaborate and prepare samples. People send us their samples. We can grow the cells and, plunge freeze them at a certain time. And then you have the remote collaboration. Ideally, though, if somebody comes to us and we teach them how to do it, we share our expertise, we share the facility, we share our consumables. And then at least you have one person that can independently go back and sort of see the technique there also. The samples can be prepared anywhere in the world, and you have cryotures, the same for crystallography, which can travel across the world and keep things frozen for, a good couple of days, 2-3 days. So that is never, well, sometimes there is, because customs sometimes can be quite weird. But trust me, we get samples easier from your side of the world than the rest of Europe right now. So yeah, so it's a bit of an investment of time if you don't have a cryogenic facility, but we either can provide our expertise or we can teach somebody, and then that somebody becomes the expert in their group.
I guess Valeria can do that.
Valeria, have you just been volunteer?
Yeah, I think so.
So I don't know if someone have more question for Maria.
Tell you what, I'll stop sharing then, so I can see you properly also.
Okay, I guess we don't have any more question. Okay, no Maria, thank you very much for your presentation, and I just want to talk with you a little more after this.
Absolutely. It was lovely meeting you guys.
Some minutes.
Yes, no, absolutely. I'll stick around.
Okay, everybody. So everybody can leave, and I will keep talking with Maria. See you.